everyone. So we had a lecture on the structure and function of DNA. We then watched a video where we learned how it's collected at crime scenes. And now that we have our DNA, what exactly happens with that DNA? What do we do with it? So today's talk is going to be about that. And what we're going to try to answer today as we kind of go over the idea of how to process DNA is we want to be able to describe the goals of PCR. We'll learn what PCR is in just 1 minute, but PCR is a big one for ultimately processing DNA and narrowing down DNA matches. We also want to be able to explain the requirements for PCR. How does it work? What type of materials do we need to run a PCR process? Third, describe the stages of PCR. There's 3 big stages where a certain set of actions occurs, and we want to be able to explain how that happens. Next, we want to go into a second process called gel electrophoresis. We want to know the goals of that process as well. We want to explain the requirements for gel electrophoresis, what's required to do it. And then lastly, describe the process of gel electrophoresis in its own steps. Now let's do a very basic review of DNA, what we've covered so far. We learned that it was a double helical structure, and we learned that there were sugar phosphate backbones, which ultimately formed the outsides of the ladder. And on the inside, we had rungs called nitrogenous bases. And those bases were made of 4 different chemicals. We had adenine, guanine, cytosine, and thymine. And we learned that as you read DNA, you do so in what's known as a 5' to 3' direction down one strand, and then that 5' to 3' direction goes the opposite way in the other one, and we call that being anti-parallel. Now, this DNA that we've learned about, understand that this is the genetic material in each one of your nuclei within your 100 trillion human cells of your body. And it's this same DNA that codes for all of your unique traits. And when we talk about forensic science, everyone has their own set of unique traits, from complete strangers all the way down to identical twins. And we'll talk a little bit more about identical twins as this course progresses. Now, ultimately, their DNA is going to be the absolute closest, nearly 100% match from one identical twin to the other. But we'll talk about some specifics that apply to identical twins as we move forward. Also, remember that we have 46 chromosomes, 2 pairs of 23, which we call being diploid. So ultimately, we have a lot of genetic material in order to look at as we try to identify different segments of DNA and how they differ from one person to another. So first, we're going to talk about PCR. PCR is known as the polymerase chain reaction. And we need PCR because when we find DNA at a crime scene, it's often in very minute quantities, a very low amount of cells containing DNA that we're going to be able to sample. So what we're going to need to do is we're going to need a process, a mechanism for copying that very, very tiny bit of DNA into a lot of DNA. And we use the PCR process to help us do that. So its purpose is to amplify a targeted sequence of DNA, like essentially putting DNA on a copier and then hitting 1 million for number of copies. So ultimately, from a very small amount of DNA, you're going to get a lot of DNA to work with. We'll be creating millions of copies of a specific piece of DNA in nothing bigger than a test tube. So think about this for a second. Imagine I had 1 piece of DNA and I need to make a lot of that DNA, and I have a process which doubles it each time it goes through a specific set of steps. So therefore, I would go from 1 to 2 to 4 to 8 to 16 to 32, 64, 128, 256, 512, 1024. Notice how quickly my numbers started increasing. So by doubling up the amount of DNA with every round of this process called the polymerase chain reaction, we can very quickly lead to very high numbers of DNA that we need to work with in the lab. So eventually, by using this as a first process, the polymerase chain reaction, it's going to allow us to go to a second process after it to detect and identify gene sequences, by using some visual techniques that are going to be based on the size of the DNA fragments. Now, that's going to be coming a little bit later, but what we want to do right now is we want to dive into this PCR process so you can see how it works. So let's talk about the requirements of this process. First of all, you can see in the picture there's 6 big things that we're going to need, plus the machine in which this process is going to happen within. One of the first most important things we're going to need is something called DNA polymerase. Now, back in biology, we learned about enzymes within the cell. Each one had its own job. Everyone had a specific task to do in terms of enzymes. And DNA polymerase, its job was to copy DNA. So we're going to need a DNA polymerase enzyme in order for us to go from a very small amount of DNA to a very large amount of DNA by copying it. And the DNA we're going to use is going to be from a hot springs bacterium. Now, that sounds kind of weird, but here's the thing about bacteria is some of them live in some very strange places, and they can withstand a wide range of environmental conditions. This particular bacterium can withstand very high temperatures, one of the things that's going to be necessary during this process. So we're going to borrow from one of our bacterial friends an enzyme called Taq polymerase. Officially, its name is Thermus aquaticus, and it can copy DNA when temperatures are very, very hot, hotter than human bodies could withstand. So we can say the Thermus aquaticus, the Taq polymerase, withstands the temperature necessary to separate double-stranded DNA. Number 2, we're going to need a supply of free nucleotides for the new complementary strand. So what's going to happen if we are creating more DNA from just a little bit? We're going to need a whole lot of A's, T's, C's, and G's to help us with that process. So we'll acquire a lot of nucleotides that we can add to our mixture so it can go ahead and start building new DNA molecules. Number 3, we're going to need little tiny pieces of DNA called primers. Primers are 10 to 20 base sequences, nitrogenous base sequences linked together, which are going to match the end of the DNA sequence of interest. So remember that DNA is a random assortment of A's, T's, G's, and C's, but we do have some known locations on DNA where certain base sequences exist. And we can build these primers because we know that they are a match for a specific sequence somewhere on a DNA strand. We're, of course, going to need a DNA sample, and because this is forensic science, typically that will come from a crime scene, a victim, or a perpetrator. Now, another couple things that you can see in our picture here is we have a mix buffer. So this is essentially where all of these ingredients are going to be mixed together. And then we have our machine itself that does the job to help us with this process. And it's called a thermal cycler. And its job is basically to send temperatures up and down and back up again in a repeated cycle, so certain events can happen at each stage. PCR does not require a lot of sample to get started, generally 1 to 1,000 nanograms, which roughly equates to about 104 to 107 molecules of DNA template. Let's do a quick overview of PCR, and we're going to say that this process can be divided into 3 general stages. Now, the first stage is going to be called denaturing. And what denaturing is generally is when something comes apart, usually biological matter, as a result of some environmental factor around it. So sometimes that could be too high a salt concentration, it could be a particular contaminant, but in this case here, it's going to be because of heat. We're going to take apart this DNA strand from 1 into 2. We're going to separate it down the middle, and you can see that now we've gone from a double-stranded, double helix portion here to 2 single strands of DNA that are now separated. Secondly, we have the step called annealing. Annealing is where we take those primers that we mentioned before, and they essentially stick to the single-stranded DNA at particular portions as almost like a beginning sequence for the copying process, which happens next. The third step is called extension, and this is where our polymerase that we mentioned earlier, our Taq polymerase, binds onto the single-stranded DNA, and free-floating nucleotides within that mixture buffer are going to come and be placed opposite to their complement. So A's will bond with T's and G's will bond with C's as this DNA polymerase or Taq polymerase moves in its forward direction down the DNA strand. So you can see here the DNA polymerase is going from a 5' to a 3' direction. This one here, same thing, the 5' end, it's building from left to right to the 3' end. And ultimately what you get at the end is you get two new strands of DNA which still have nucleotides from the original parent strand. Let's take a quick closer look at that denaturing step. So in the first stage, your double-stranded DNA will be separated into two single strands by breaking the hydrogen bonds between the nucleotide base pairs. So if you can remember, we talked about down the center of those nitrogenous bases, or the rungs of our twisted ladder, hydrogen bonds hold these bases together. By increasing the temperature, what's going to happen is it's going to cause enough vibration for the two sides to come apart from one another. In other words, to break that hydrogen bond in the center. So it typically happens around 94 degrees Celsius. Now remember, human body temp operates at 37 degrees Celsius. So ultimately, our DNA polymerase would come apart at temperatures that high. But we borrowed our polymerase from Taq or Thermus aquaticus in order to help us with this process. So that machine that we mentioned earlier, the thermocycler, will heat up the mixture in the vial to 94 degrees Celsius. To fully denature DNA, it takes only about 10 to 60 seconds, about 30 seconds on average, to go from one strand to two. Once this step is complete, continuation through the next two steps called annealing and extension can begin. During the annealing step, primers, those short sequences of known DNA, are going to bind to complementary sequences of the denatured DNA strands. So in other words, this primer right here has been geared to bond to these four bases right here. Same thing with this one here. These will be put in the mixture. They're going to find their complementary binding sites, bind onto it, and ultimately give our Taq polymerase a place to begin its process. So how do we get this to happen? Well, remember the thermocycler is a machine that raises the temperature and decreases the temperature pretty rapidly. So ultimately what's going to happen is the thermocycler will take the temperature back down to 52 to 58 degrees Celsius, and the annealing time will be maintained for about 15 to 60 seconds. So ultimately, by dropping the temperature down below, we're able to allow these primers to adhere or stick to their complementary base pairs in order to reach the next step. Our last step is going to be called extension. So extension is the final stage of the cycle, and it occurs at a temperature that allows for optimal polymerase activity of binding nucleotides to the annealed primer, resulting in the exponential amplification of the template strand. Whoa, that's pretty heavy. What do we mean here? Well, remember, we talked about the Taq polymerase, which is this green C-shaped blob right here. It's going to need to bind onto the end of the primer, which is going to happen right here. Now, when it does so, it's going to start moving in the direction from right to left in the case of this one here. And as it moves from right to left, it's going to be adding these single nitrogenous bases to their complements that are exposed on this top strand. So this parent strand up here is going to be the template for this Taq polymerase to bind its complement at every single nucleotide, every single nitrogenous base that you see down the center. Meanwhile, another DNA polymerase is going to be working on the other strand that got separated. The parent strand is in blue, and this one is going to move from left to right, and it's going to add these single nucleotides to their complements as it moves from left to right. At the end, what we're going to get is we're going to get two new strands composed of the old parent strand, and the other side will be the new composed strand from what was in the buffer mixture. For this last step in the process, we got to go ahead and change the temperature one more time. So we started off high in step 1, denaturing. We took it down a little bit during the annealing step, and now we're going to go back up a little bit, 70 to 80 degrees, which is going to be optimal for this particular polymerase to operate for about 1 to 2 minutes. And that's all you're going to need because this happens really fast. And ultimately what we just did was we went from 1 strand to 2. Now, understand, we now have 2 strands. We're nowhere near the amount of DNA we're going to need to test in a laboratory. So we're going to have to run this over and over and over again, and it's going to go through these 3 cycles, and it can go anywhere from an hour to a day, depending on how much DNA that you need. Now remember, we're going to need a lot of DNA to work with, millions and millions of copies. So we're going to have to run through this procedure, run that thermocycler for a number of hours until we get enough to put to work and analyze in a forensic lab. I'd like you to go to www.sumanisinc.com, click on animations, find molecular biology, and then click on PCR to watch this process in action. So now we've run our DNA through the PCR process. We've gone from just a few copies of DNA to millions and millions of copies of DNA. So what happens next? Our next process is called gel electrophoresis. This is going to be the technique of using DNA fragment lengths called RFLPs to match and investigate DNA samples. Now, how is this done? What we're going to do is we're going to treat DNA samples with something called restriction enzymes. So remember, enzymes were these little protein helpers within cells that did specific jobs. Restriction enzymes are going to kind of work like chemical scissors. They're going to make cuts at specific places. And by doing that, we'll ultimately produce a unique collection of different length fragments. Electrophoresis of those fragments that are cut will separate fragments according to their charge and their size. This process will also be used to measure the number of repeats of short DNA sequences that are unique to an individual. So remember the end goal here. We're trying to determine how one person's, whether they're victim or perpetrator, DNA can be connected to a crime scene or to each other. When we do this, it will produce a unique, very distinctive banding pattern. If you look down here in this picture, you can see that there's almost what looks like a barcode for these letters here. We'll go into what that means in the slides ahead. Let's bring to the forefront the idea of these restriction enzymes and their job. It's going to be really important because ultimately it's because of them that we're going to be able to analyze DNA from one person to another or one person to a crime scene and see the differences that exist there. So ultimately, the job of a restriction enzyme is like chemical scissors. It's going to be cutting DNA at very specific places. So if you look at the example below, we can see the DNA in this 9-base segment right here with its complementary strand on the other side. Now, if we go ahead and we hit that with restriction enzymes, you can see that it's going to make specific cuts. And the cuts that it's going to make are right here between this A and this T, this A and this T, this T and this A, this T and this A. And ultimately it took this longer segment here and cut it into shorter segments with what we know as sticky ends. So sticky ends are basically where our double strand goes to a short little addition that is single stranded, but ultimately this longer strand of DNA got cut into 2 pieces that are now going to be shorter than the original one was before. Let's take a look at the basics of electrophoresis. So electrophoresis is what we do in lab for separating mixtures of charged molecules. Remember from back in the day what a mixture was. It was a material composed of 2 or more elements or parts. And some of those elements and parts can be charged molecules. A molecule such as protein or a nucleic acid like DNA that has too many or too few electrons. If you have too few, you wind up being positive, and if you have more electrons, you become negatively charged. And you can see our little sample jar here, a sample container. We have a number of different particles, red, blue, green, and yellow, and some of them are charged positive and some are charged negative. They're a mixture. Now, charged molecules can be separated based on 2 things, electrical charge and their size. So if we take a look over here, we have this random cluster of blobs of beige and red. How could we separate these molecules from one another? Well, first, if we were talking about electrical charge, hopefully you could hypothesized that we could use the opposite charge in order to get half of them one direction, half of them the other way. So if I had, for example, some kind of negative electrode or negative plate, I might be able to pull out these positive molecules, the beige ones. And if I had a positive plate, I could be able to pull out the negative molecules because, as we know in science, that opposites attract. So now we have two piles of molecules, half of them charged positive, half of them charged negative. What could you do next to separate them based on size? What if we had a series of strainers with different pore sizes? Some of the pores are large that allow most of the particles through except for the biggest ones. The next level down would be somewhere in the middle, and it would allow the two smallest particles to get through while trapping the two largest molecules. Go down pore size one more time, and then the next largest size will get caught in this sieve or this porous strainer. And then you're down to 4 separate groups based on filtering through a number of different pore sizes. And you could do that for both the positive and the negative molecules. Gel electrophoresis also has some essential parts in order for it to work properly. We're going to need electrical current during this process to ultimately use that electric current to force particles from one side of a sample to the other. Technically, electrical current then is going to be the flow of electrical charge from a negative pole to a positive pole. The negative electrode or pole, we're going to call that the cathode. And in this case, it's going to be coming from a wire that emits electrons. The other side will be an anode or positive electrons, a positive electrode, which will be a wire that collects electrons at the other side. Since it's called gel electrophoresis, there's a good chance there's going to be a gel in it. That gel happens to be called agarose gel, and that's going to be what we call a porous medium. So it has holes within it of different size. So think of the last time you ate Jell-O. Imagine that within that Jell-O, that there are tiny little pores, tiny little holes that things could essentially move through. And that's kind of the idea of what an agarose gel does. It's permeable to fluids. It allows fluids to flow through it and very small particles, but it can filter or catch larger particles as they try to move through. The pore size is very small. A buffer fluid will be poured over the whole sample in order to allow this process to continue because it'd be very hard to get this stuff fly through air. So we have it all in a liquid medium in order to control the flow of these molecules. All of these will be found in what's known as an electrophoresis unit. Now, separation of our DNA from its initial location to a future location on our gel is going to occur because of two things. One is going to be electrical charge, the other we'll get to in a minute. So, understand that within the body, there are different biomolecules charged positive and some are charged negative. A lot of molecules have naturally occurring negative and positive charges on them. Amino acids, peptides, proteins, DNA, and RNA. And the sum of the charges determine the overall charge. So if we look at some of the molecules below, you can see, for example, down here, this is a positively charged amino acid. You can see the positive right here. You don't see a whole lot of other charges shown in this case. And this peptide right here, you can see 2 negative charges and you can see 1, 2, 3, 4 positive charges. So overall, this molecule will be charged positively. Over here, our last one, you can see 1, 2, 3, 4, 5, 6, 7 positive charges. And we have 1, 2, 3, 4, 5, 6, 7, 8, 9, a whole bunch more negative charges. So overall, that last one is going to be a negatively charged protein because there's more negative charges exposed than positive charges. Now, when any biomolecule is going to be exposed to an electrical current, what will happen is ultimately wherever the flow of electrons is going, and in this case it's going to be happening from the negative pole to the positive pole. If you're charged positively, you're going to stick around close to the negative electrode. If you're charged negatively, you're going to be repelled by a flow of electrons in the opposite direction. So ultimately here, if we had a negative electrode, which is emitting electrons, the positive molecules would tend to hang out because opposites attract. And meanwhile here, this negatively charged protein would be repelled by a flow of electrons, try to get away from it because like charges repel. So ultimately the movement The moving of the negatively charged particles is going to be based on charge. But where it truly gets separated is going to be through the gel or the porous material that it's flowing through. So remember, there are little tiny microscopic tunnels and holes for particles to flow through. But the key is to get through them, you got to be really small as well. So small molecules will move a lot faster than large molecules and get further within the gel itself. So if you can imagine this here as the inside of one of these gels, you could see that large particles would have a hard time getting through, mid-sized particles would wind up somewhere in the middle, and the smallest, most negative particles, those would be traveling the furthest within your gel. So this is what a basic gel electrophoresis unit looks like. You can see this is nothing more than an electrical box. You can see the cathode on the left-hand side, and this is a wire that basically drops down into a fluid here, and it's negative in nature, which means it emits electrons from it. And over here we have a positively charged pole. This is your anode, and this is going to collect those electrons that go from left to right over here, okay? Now, over here, this is going to be your gel. A gel made of agarose is kind of, it's a jelly-like, but a little bit thicker and a little bit tougher than Jello or a gelatin. It holds together reasonably well. And what you see here are these little rectangles. These are depressions within the gel where we add our DNA samples. So all of this is under a liquid called a buffer. So what's going to happen is when we have different samples of DNA, we're going to use a tool called a micropipetter. And what we're going to do is we're going to extract some of this DNA and we're going to put it down in this well. Now, this takes some practice. It's a little handheld device. It takes very minute quantities of samples and allows you to inject them into these very small depressions in a gel underwater. So it definitely takes a little bit of practice to get good at it, but after a few times, most people can do pretty well with it. So again, the DNA gets injected into these here. And what's going to happen is once they're all injected in these wells, after a very short time after flipping on this electrophoresis unit, what's going to happen are those negatively charged DNA molecules. Remember, some of our biomolecules are negative in nature. DNA is one of those. And it's going to begin to move down through the gel from left to right. So after a short time, what will happen is that DNA will start moving in that left to right direction. And when we hit the off button on our gel electrophoresis unit, everything will freeze in place. So after a given time, we'll say a half hour or so, we shut off our gel electrophoresis unit, and then our DNA has moved. Some of it has moved away from the gel, not very far at all, and these would be your longer molecules. Remember, they're going to get caught because they're just too big to move through those tiny little microscopic tunnels. We have some in the middle here, so they're going to be mid-range in size for DNA fragments. And then down here, we have the shortest molecules of all. They're going to get the furthest. Now, how did we get to all of these fragments of DNA? Remember that with our DNA samples, we add restriction enzymes to them that cut them in specific places. So ultimately what we have is we have a vial here of DNA that's made of very small segments of DNA, medium-sized fragments, and large fragments as well, based on what fragments are left over after a restriction enzyme makes its cut of those samples. If you get a chance, Google NOVA, create a DNA fingerprint, and that will allow you to do a virtual DNA fingerprinting exercise. It will take you through the steps so you understand how this process works one step at a time. So what we're doing in gel electrophoresis is we're doing restriction fragment analysis. We get a restriction fragment from ultimately mixing our DNA sample with restriction enzymes. That's going to produce fragments. So once we have fragmented two different samples of DNA with the same restriction enzymes, we can then compare those two to see if they broke up in the same way and if they have the exact same DNA fragment lengths or not. So in this example here, we have two different DNA segments. This one is called a normal beta globin allele, and this one down here is called the sickle cell mutant beta globin allele. Both of them are fragments of DNA of a certain length. Okay. Now, what we're going to do is we're going to hit them with a restriction enzyme called DDE1. Okay. So DDE1 is going to break up this fragment here. It's going to find 1, 2, 3, 4 places to cut this fragment because it found the specific letters it was looking for. And when it did, it cut them in those locations. In this second strip of DNA, notice that DDE1 cuts it in 1, 2, 3 different locations. Now, what does that mean? Well, if we look at the gel electrophoresis that was done by ultimately inserting both of those samples of DNA, this one here went to this well, and this one here went to this well. We turn on the gel electrophoresis unit. We shut it off after a given amount of time. And what we found was we found 3 different fragments for this strip of DNA right here. And if you look at the 3 fragments that it found and that it separated, it was this one here, and this one here. Okay? So ultimately, remember that large fragments get caught up early in a sample a lot quicker than short or even medium ones. So as we turned on the gel electrophoresis unit, the DNA, all of it started moving this way, but this big large fragment right here got caught up right here very early. As it progressed, the remaining 2 fragments, one of them was 201 base pairs and the other one was 175 base pairs. That's what these 2 bands are right here. We almost like froze them in position when we shut off the gel electrophoresis unit. Meanwhile, on this particular strip of DNA right here, you'll notice that we have, again, a large fragment with exactly the same cuts as we saw in the one above it. So that large fragment also got caught up in the same exact location here as the other strip right there. But you'll notice here we didn't get this middle cut right here. You don't see it at all. You do see the one here on the left. You see this one here in the middle, but it's missing this one here. And that's because this strip of DNA is slightly different from that one. There's different bases in this region right here, which allows no cut to happen because that restriction enzyme did not recognize the specific set of letters it was looking for at this position. So as this one continued on, the remainder of the DNA got caught up at 376. So you can see clearly these 2 strips of DNA are not identical. This one has 1, 2, 3 different clumps of DNA that got stuck, and they happen to be at different locations than the 2 that you see here. This one here does match, but this one here has no match in this strand, and these 2 clumps of DNA have no match in this strand here. And that would tell someone visually observing these 2 strands of DNA that they are not identical to one another. Imagine there was a child in question during a paternity test, and we were trying to find out who was the true father of that child. What we could do is we could take DNA samples from all 4 people, the mother, the child, and the 2 potential fathers, run it through gel electrophoresis, and see how many bands match from each of the fathers to the child so we could determine paternity of that child. Take a look at Mother's bands. Mother's bands are all purple, and you can see that a lot of her bands match with this child in question. Now, if we take a look at the 2 fathers who have red bands, what we would be looking for is from the divided up DNA that gel electrophoresis provided us, which one matches more bands that we find in the child. And you can see right here, we have a couple of bands right here that are a perfect match. We have these right here. We have these here as well. But if we take a look at the male number 2's bands, you can see there's an absent spot right here where we see one in male 2. We see nothing here as well, nothing here, nothing here. So there are a lot more bands correspond with male 1 than we see with male 2. So that would allow us to believe that male 1 was the father with the greater amount of certainty than male 2 if it was just in question between these 2 individuals. If you get a chance, please Google McGraw-Hill DNA fingerprinting for a very nice video that represents this entire process. Also, go to sumanisinc.com, click on animations, biotechnology, and go to paternity testing for an even greater in-depth view of this process. The current strategy for differentiating one individual from another is going to be based on something called STRs, short tandem repeats. Now, STR analysis is going to use loci in DNA where the same sequences of bases may occur a number of times. Now, a locus on a chromosome is just a location where we find something. So all of these yellow boxes that you see right here, these are locations of regions where we find these STRs, or short tandem repeats. So ultimately, a short tandem repeat is an area on the DNA that's known to have very short, repeated sequences of letters over and over, sometimes 5 times, sometimes 10 times, sometimes 20 times in different individuals. Imagine we have 2 individuals. We have person 1 and we have person 2. And at the end of a sequence of letters, we find a sequence that begins G-A-C-T, 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 over and over and over again. Okay, so in this person here, let's just keep it going a little bit. G-A-C-T, and one more time, G-A-C-T. Okay, we have 1, 2, 3, 4, 5, 6, 7 G-A-C-T repeat short tandem repeats, we call those. In my other individual, in the middle of their DNA, they also have that repeating sequence of letters, G-A-C-T, but theirs only goes 4 times. So this person here has a much longer segment of this repeating unit called an STR, and this person's right here is much shorter. So when you hit these chromosomes with restriction enzymes, it starts cutting them up. What's going to happen is individual number 1 is going to have longer segments connected to that STR than individual number 2. So ultimately, if we do this a whole bunch of times, where you see these yellow boxes on the different chromosomes, that's going to allow us to have different lengths of DNA that we're then able to run through a gel electrophoresis process in order to separate them and see them. So ultimately, we use this process to differentiate one individual from another. The FBI is going to use 13 known locations on chromosomes in order to do this. The possibility of 2 individuals coding the same in STR for all 13 of those loci is greater than 1 in a billion. So when that particular case comes to court and they talk about what is the possibility of it being from any other person or place, and the number of 1 in a billion enters the courtroom, that's pretty heavy for a jury to hear and ultimately make their decision based on. That will conclude today's lesson on PCR and gel electrophoresis. Now, remember, PCR and gel electrophoresis are 2 parts of a bigger process called DNA fingerprinting. So if you look over here at these samples that you can see on this gel, you can see that each individual here, this mother, this child, and this father, they all have their own unique DNA fingerprint. And that's ultimately what PCR and gel electrophoresis give you, is a fingerprint of an individual whose own DNA segments and bands are completely their own. Again, with the exception of identical twins, that everyone who's different on the planet is going to have a different barcode of DNA segments that make up the DNA inside of their own personal genome. So what should we remember from today's talk? Well, obviously, we want to know what were the goals of PCR. Why do we use it? Second, what were the requirements for PCR? What were all those little ingredients and parts for? What did they do for the process? What were the stages of PCR? Describe the goals of gel electrophoresis. What were we trying to do with those DNA samples? What were the requirements for gel electrophoresis? Why do we need what we need for that process to run correctly? And describe the process of gel electrophoresis in the different steps. Hopefully you understand the process of DNA fingerprinting a little bit better now. You can see it has nothing to do with actual fingerprints, but instead it has to do with someone's DNA and how it separates. More to come on these topics. Stay safe, and I'll see you next time.